Hello and welcome. Okay, let's just jump right in because today we're tackling one of the all-time classic points of confusion in biology, the difference between Mendel's law of segregation and his law of independent assortment. Seriously, if you've ever felt your brain tie itself in a knot over these two, trust me, you are in very, very good company.
And hey, don't just take my word for it. This exact question pops up constantly. I mean, look at this post from a student on Reddit. This is a real thing people struggle with. So today, we're going to untangle this thing for good. And right here, this is the heart of the problem. You look at the definitions side by side and they sound so similar. They're both about stuff separating when you make sperm or eggs. But here's the key difference in a nutshell. Segregation is all about the two copies for one single gene. Independent assortment, that's about how different genes for totally different traits behave with respect to each other. To really get that, though, we've got to go back to the beginning. So here's our game plan for this explainer. We're going to start with the man himself, Mendel, and his famous peas. Then we'll break down the law of segregation, then the law of independent assortment. After that, and this is the cool part, we'll connect his ideas to the actual chromosomes that he never even knew existed. And finally, we'll see why any of this still matters today. Okay, let's set the scene. Picture it. It's the 1850s. We're in a monastery garden. Nobody knows what DNA is. Nobody's ever heard of a chromosome. And this monk, Gregor Mendel, is about to fundamentally change science with basically some pea plants, a little paintbrush, and an incredibly organized mind.
This was his mission, in his own words. I mean, for centuries, people knew that kids looked like their parents, but the rules were a complete mystery. It just seemed random, messy. Mendel was looking for the pattern, the instruction manual that governed the whole thing. Now this right here, this shows you his genius. It wasn't just what he found, it was how he set it up. He was incredibly methodical. He picked 7 very simple either-or traits. The plant was either tall or short. The seeds were either round or wrinkled. There was no in between. This simple binary approach was the key to everything. And get this, he came up with a brilliant way to track these traits. He didn't know about genes, so he just called them factors. And he proposed that for every trait, a plant had 2 of these factors. He used a simple code, a capital letter for a dominant factor, the one that always shows up, and a lowercase letter for a recessive one, the one that can hide. This simple idea was revolutionary. So let's see how he used this to make his first huge breakthrough. To start, he kept it simple. He focused on just one trait at a time, like, say, how tall a plant is. He'd cross a pure tall plant with a pure short plant. The big question was, what are the kids going to look like? Well, he found that all the offspring were tall. The short factor seemed to have just vanished. But, and here's the kicker, when he bred those hybrid plants with each other, shortness came back. This led him to his first law. The 2 factors for a trait must separate, or segregate, when you make reproductive cells. Each sperm or egg only gets one of the 2 copies. But here it is, the crucial piece of evidence, the smoking gun. It wasn't just that the short trait reappeared, it came back in this super specific, predictable way. For every 3 tall plants, there was exactly 1 short plant, a perfect 3-to-1 ratio. And he found this exact same ratio for all 7 traits he looked at. That just couldn't be a coincidence. And Mendel's true genius was realizing that this 3-to-1 ratio that he could see was actually hiding a deeper 1-to-2-to-1 ratio in the genes, or factors. So of those 3 tall plants, 1 was pure tall, but 2 were hybrids that secretly carried that short factor. And of course, the 1 short plant was pure recessive. This proved it. The factors were separate little packages that split up and then recombined. Okay, so that's segregation. The 2 copies of 1 gene separate. But Mendel, he was just getting started. He then asked a way more complicated question. What happens if you track 2 different traits at the same time? Like, does a seed that gets the yellow factor also have to get the round factor? Or are they totally separate deals? And this led to his second great law, the law of independent assortment. And it basically says that genes for different traits are sorted into gametes independently. The allele a gamete gets for seed shape has absolutely 0 influence on which allele it gets for seed color. They're free agents. So to test this, he did what we now call a dihybrid cross. He started by crossing a plant that was pure for both dominant traits, round and yellow seeds, with a plant that was pure for both recessive traits, wrinkled and green.
The first generation of kids, as you see there, were all hybrids, showing both dominant traits. But the real test, the moment of truth, was what happened when he crossed those hybrid kids together. And boom, look at this. When he counted thousands of seeds from that second generation, he didn't just get the two original combinations. He got 4 different types, including brand new ones, round seeds that were green and wrinkled seeds that were yellow. And they always appeared in this beautiful, consistent 9:3 to 3:1 ratio. This was the proof. This could only happen if the factors for shape and color were assorting independently. So Mendel had figured out the mathematical rules of the game. He basically wrote the book on heredity. But he had no idea what the physical game pieces were. He never saw a gene. He never saw a chromosome. The why behind his laws was a total black box that he, sadly, never got to see opened. And it took a long, long time. He published his work in 1865, and for 35 years, crickets, nothing. It was basically ignored. It wasn't until 1900 that a few scientists rediscovered his paper. And just 3 years after that, 2 guys, Sutton and Bovary, who were actually looking at cells under microscopes, had the ultimate aha moment. They realized that the way chromosomes behave during meiosis, that's the cell division for making sperm and eggs, was a perfect physical explanation for Mendel's laws. It was uncanny. Mendel said factors come in pairs, while chromosomes come in pairs. Mendel said factors segregate, while chromosomes segregate. Mendel said different factors assort independently, different chromosome pairs assort independently. It was a perfect match. So let's use this image to really nail down the difference. Look at the diagram on the left. Just focus on that one pair of chromosomes. The law of segregation is about how that one pair separates, so each gamete gets either the big letter allele or the little letter allele. Now, look at the diagram on the right. The law of independent assortment is about how the separation of that first pair of chromosomes has absolutely no influence on how the second pair of chromosomes separates. They do their own thing. That's it. So why do we still care so much about this discovery from over 150 years ago? Because these aren't just rules for pea plants. These are the fundamental rules for us, for animals, for crops, and they have massive implications for our health and our future. But there's a really cool plot twist. It turns out Mendel got a little bit lucky. All 7 of the traits he picked just happened to be on different chromosomes. We now know that if genes are physically located close together on the same chromosome, they're linked. They tend to get inherited together as a block. And the wild part is, discovering this exception, the thing that broke Mendel's second law, is actually what helped prove the whole chromosomal theory was correct. And this visual makes it super clear. On the left, independent assortment. The genes are on different chromosomes. Think of them as being in different cars. They can go to different destinations. But on the right, that's linkage. The genes are passengers in the same car. Where one goes, the other has to follow. Understanding this difference is absolutely critical in genetics today. And the legacy of these laws is just everywhere. In medicine, we use them to diagnose conditions like cystic fibrosis. Genetic counselors use them to help families understand their risks. In agriculture, we use them to breed crops that can feed the planet. I mean, even the most cutting-edge technologies like CRISPR gene editing are all built on the foundation that this monk laid in his garden. So I want to leave you with this thought. Mendel saw a perfect mathematical pattern and figured out the rules decades before anyone saw the machinery behind it. It just makes you wonder, doesn't it? What fundamental patterns are we observing today, right now, without having any clue what the mechanism behind them really is? Thanks for joining this explainer.